I'll give you a little overview of what we do in my lab. My lab is called the Microsystems and Nanosystems Laboratory. And the idea there is really very simple. We want to use tools of micro and nanoscale technologies to develop various kinds of systems, which means devices that have the ability to perform more than one operation on the device itself, and then address a variety of problems. Now, you can probably see from this slide, I wear a fair few hats in our department and around the university, which is pretty exciting because that means that I get to collaborate with lots of different people, learn from lots of different domains, and that has been a very valuable experience in my 10 years or so at Ohio State so far. So, briefly what we do, growing up, I heard about Atlas carrying the weight of the world on his shoulders. And as sort of the modern society grows, I realized there's sort of, at least for me, there's not just one world, there are three worlds that Atlas carries on his shoulders. One is where we all need clean water for a variety of industrial and health applications. There's obviously the healthcare domain that we all worry about, especially in the present climate. And then as engineers, we also worry about the energy portion of this puzzle. And these three domains intersect in different ways. So where do we come in? So this is me and perhaps a representation of my handsome self. I know my students probably don't want to be characterized as Fred Flintstone, so I'll leave them out of this one. But we come in with our tools of micro and nanotechnology to chip away at these problems. So what we really do is we develop what I call functional device solutions or technology-based solutions. And they have sort of two prongs that we address. One is to try and understand what is happening as these nanoscale or microscale systems interact with whatever application space we are in. So there is a fair bit of basic science we do. And there is also a translational part of that, and I'll talk about that in a little bit, but we sort of like to see our devices getting used in the real world. So there is sort of a iterative path between the basic science and technology components that we do. So what does that mean? To solve these problems in healthcare, energy, water treatment, my group generally requires people who have a really strong interest in interdisciplinary and perhaps multidisciplinary problems as well. So what do I want to do today? I want to talk to you about just 3 examples of ongoing work in my lab where we use these tools of micro and nanoscale systems to target 1 application in energy and 2 in healthcare. That's sort of the overview I'll present. Because of time, I don't think we'll be able to get into the water side of things and maybe some of the other energy projects. So I'll sort of limit myself to just 3 examples. So the first example I wanted to talk about is that of chronic wounds. So just to put things in perspective, chronic wounds could be something like a diabetic ulcer. It could be somebody who suffered a burn injury that has not been healing because of severe damage to tissue. And management of chronic wounds in the United States alone to our healthcare system costs about $25 billion a year. And at present, there are approximately 6 to 7 million people in the United States who deal with chronic injuries of some kind. These are dermal injuries, typically on the limbs or extremities, maybe other parts of the body. This could be post-surgery. The management is pretty difficult, multiple visits to the doctor's office, expensive. So, what makes wound management so hard is that wound healing in itself is a multi-stage process. So, the hemostasis or the homeostasis is the first step in this multi-stage process where basically post an injury blood flow will stop and blood clots form. After that, we get into this inflammatory stage where, if you've had a little cut, you've seen the cut turn sort of reddish, pinkish in color on the outside. That's your body responding and putting together an inflammation path to initiate healing. And then it gets into the idea of bringing in other healing cells that start to bring in the epithelial or the skin layer to close out the wound, and then finally tissue remodeling to try and restore the function that was lost at the very first stage when the injury was caused. So the idea here is that, biology sort of works on a trial and error basis, right? So once the injury occurs, biology kicks in and it is trying to figure out what is happening, and it sort of goes through these processes without any specific systematic control on it. Moreover, each of these stages is susceptible to infection. So if you have a wound that will not heal, usually it leads to fairly nasty infections in the form of bacterial biofilms. So these are, ooey gooey substances that get collected on the wounds. And, for people who don't like to see this, I'll tell you in a slide or two, we're going to see some pretty gross images. So, as a mechanical engineer, I tried to stay away from that for as long as I could, but, eventually I got pulled in. So, if you don't want to see that, I'm giving you a heads up here, please, stay away from the screen. So the problem with infections is they add a complicating factor in terms of wound healing. So they hinder the natural process of healings if there is infection present. Now, there are lots of different ways people try to control infection. It could be anything from physically scraping it off, called debridement, to use of antibiotics and other antimicrobials, negative pressure therapy. There's all sorts of tools that are available right now. Similarly, there are lots of approaches to tissue regeneration, but at present, there is no way to control scar tissue. So if anybody has old scars from various kinds of injuries, that it's just the healing process, and being able to heal tissue without causing scars is a very important process, because scar tissue can lead to either incomplete healing, incomplete healing, or the function of the final tissue may not be fully restored. So here's our solution, right? So given these problems, my lab came up with this idea of what we call speeds, because we do find that it heals wounds faster. And what it is it's a smart platform because we have several sensing capabilities on it, and it's an electroceutical dressing. So electroceutical, analogous to pharmaceutical, it uses electrical signals instead of any kind of pharma-based or drug-based approaches to heal wounds. What you're seeing in the picture here is the back of a pig. So this is a in-lab experiment where we induce 6 wounds in the back of a pig, and the yellowish substance you see is a very serious infection that we introduce on the back of a pig. So if we zoom in, this is what it looks like. These are representative of what serious infections in humans might look like as well. If you put this through an electroceutical treatment with our dressing, you can see a picture of our screen printed dressing. This is on silk. So it's a fabric-based dressing, which is very flexible with a flexible battery pack and everything that we integrate with this. The tissue looks a lot better. Sort of the pinkish hue, as I've been told by clinicians, is a mark of a tissue that's beginning to heal. So we could quantify this, and what it showed us was, if you look at this plot here, the y-axis showing sort of the reformation of the skin over a period of few days, and consistently the green bars, which were our electroceutical treatment compared to the state-of-the-art sort of dosing mechanism that we've used, we find that the electroceutical does better than any other treatment method that's out there. And it does better by about 30% on average in terms of wound closure. We have as everybody does, there's a lot of these papers that have come out in the last couple of years on this topic. So the big question then that comes up is, if you have this in the lab, it works, you have this nice dressing, would it actually work in a clinic? So, being at Ohio State, one of the opportunities we have is to collaborate with a fantastic medical school and also our partners in the veterinary school. And, they contacted us about 2 years ago or so, 2.5 years ago or so, to talk about a dog. And, there's been a bunch of press reports on this. I won't get into that, but this dog had an injury on its front left paw. It's a 4-year-old dog. And, it had been injured for about a year or so, and they'd gone through a series of what you would call state-of-the-art treatment, starting with negative pressure therapy, skin grafts, antimicrobials, heavy pain medication, and the wound just wouldn't heal. So now I'm going to show you some of the pictures on what happened to that dog. So, when we first started on this, you can see this injury on the left corner in the first panel, which was the wound there, and it just wouldn't heal. So, we started to treat the dog and went through about 10 or 11 days of treatment with our PED. And this was a dog that came to Ohio State from Cincinnati, had been in the hospital for a while, and the costs were too high. So, about in 11 days, when the wound looked like it was healing, this patient was discharged from the hospital, sent back, and they brought back the dog almost 2 months later for the inspection. And you can see the wound is fully healed, and there was hair growth. So, we couldn't track the dog during that 2-month window, but there was no treatment. It was just left open and let it heal on its own. So it does work in the clinic. Since then, we've done these tests on another dog and a cat, and I'm proud to report that all of these the 3 animals are healthy. We've also done some efficacy testing on humans with diabetic ulcers, and this thing seems to work. So it's exciting. So this is the translational part of our work that we get to see from sort of conceptualizing the design, building it, and taking it all the way to the clinic. The second example I'll talk about is, and I'll go a little bit faster because I realize we may be getting close in time. The second example is that of, sort of a different domain in my lab where we use small scale fluid flow to evaluate how infectious diseases might work. So this, comes out of a different project in collaboration with John Song, but that's looking at cardiovascular disease. But now we are developing these microfluidic devices to look at viral vectors. And specifically, we are looking at how viral vectors can impact the risk for cardiovascular disease. So, it's been known that HIV is a known marker for enhanced risk in patients for cardiovascular disease and serious cardiovascular events. There are recent emerging reports that also identify COVID-19 in the same domain. Significant blood clots and cardiovascular events have been reported in people with COVID-19. So, our model works in that domain as well. So there was really a need for a technology that can be developed and reported on to understand how blood flow in vessels typically impacts cardiovascular disease, and if there is a way to developing new therapeutic interventions for that. So, a few years ago, in collaboration with John's group here in mechanical engineering, we developed this sort of what we call a vessel bifurcation system, where the flow comes into the device, to this device from this bottom side, divides into 2 channels. So it behaves as if it's a splitting blood vessel. And in the middle, we have a pocket where we sort of have tissue mimic collagen here. And sort of these open windows that you see where my mouse is pointing, in the branching vessels or in the bifurcation point, we seed cells that would be representative of blood vessels. And we look at the interaction between flow and the cells, and as a combination of stagnation pressure, which is impinging at this point, and then shear stress as the flow goes parallel to the cells. And what we found was the real advantage of this device is that, apart from capturing the geometry and the blood vessel tissue interface, we are also able to put in a sort of variety of stimuli, could be mechanical through flow, chemical stimuli through various kinds of drug or other antigens or infection agents, and then electrical stimuli should be needed for therapeutic reasons. Very quickly, what we found in our study when we spiked the incoming flow with blood serum or ingredients from HIV patients is that, in HIV patients, the leakiness of or the permeability of the blood vessels goes up. And this is what causes serious heart events or cardiovascular events. And this is sort of similar to the mechanisms that are being seen with COVID-19. The last example I'll talk about is sort of this different project which sits in the energy domain. And it's sort of an exciting project for us to do nanoscale material assembly, especially if we can get to the tunable optical properties part of this. And really the idea here is that, can we modulate the energy coming into buildings with light from the windows, have them sort of allow more or less light to modulate heating, cooling, and light loads. And that would be a big deal to do. The challenge really here to perform this kind of an operation is to assemble nanoscale materials, typically with critical dimensions less than 500 nanometers, in an organized manner. And the reason for this dimension of 500 nanometers is sort of trying to pick on visible light or near infrared light. So, we've developed this system where we have a print head that we've developed using microfluidics, and it uses a combination of, regular pressure-driven flow with an electrically driven flow. And the upshot here is we are able to print some really interesting nanoscale particle structures onto a variety of substrates. And in this first image, you see just rows of lines being printed. You take a closer look through an electron micrograph on these images, you see that these rows that look really straight in optical micrographs have a little bit of a fine structure to them that still needs to be optimized. We're working towards that. Or we can also print grids. So now this is the first attempt to try and get structures that can allow light to go in X and Y directions. The Z direction is still something, or normal to the page is something that we're still working on. The advantage of this kind of process really is that compared to any of the other methods that that exist right now, our printing method is about 100 times faster than anything that's out there right now. So, I know I rushed through a couple of these last examples, but that's where I'll stop. But I do want to sort of thank you all for, taking these few minutes to listen to me. I'd be remiss if I didn't thank everybody who's in my group, because none of these experiments and wonderful pictures would be possible without all the folks here. Our group is a very collaborative group, both within mechanical engineering and across the medical school and veterinary sciences and microbiology. And of course, we're very thankful to our industrial and federal partners for, sharing the gold with us, because without that, none of this happens. I do have a link here for my website or our group's website and my email if people have other questions. And I don't know how much time we have, Nick, but perhaps we could take a handful of questions.
So we'll open it up for any questions that there are from the audience right now. We still have a couple of minutes, so you're perfectly fine time-wise. I'll ask a question while we wait to see if anything comes in. Did you anticipate, you would be doing this line of research, when you started as a faculty member? Did you see kind of the variety that you're into?
Well, now that I have tenure, I can answer it truthfully and say probably. When I interviewed at Ohio State, I presented a much more narrower path and more, specific path to what the line of research would be. The one thing that has been a little bit of a surprise is sort of the investment we've had in the last, I'd say, 5 years on the bio side of things. Again, fairly truthfully, I can admit that I never envisioned or imagined myself getting this heavily involved on the bio side of things and or the biology side of things. And really, what pulled me in there was sort of this interaction between infectious disease and sort of the role that as engineers we can play in not only understanding the fundamental mechanisms, especially coming from the fluid mechanics side, for how these diseases progress, spread, impact the human and animal bodies, perhaps even on the plant side, and then develop tools to look into that. So that was a little bit of a surprise to myself, I guess. But I think the potential had always been there with these micro and nanoscale technologies to be a very broad area of research.
So then the follow-up question I'm going to ask is, given the current situation we're in with COVID-19, what are some of the challenges that you've seen, but also, where do you see your research and see the research in general going after kind of this experience?
Well, I mean, COVID-19 has been, I'd say, a very difficult pandemic. Part of it is that the sort of the mental health and the physical toll it takes on people is pretty serious. And, with the shutdowns, being out of labs, not being able to see students that I meet weekly, sometimes multiple times a week, not meeting colleagues and not just walking down hallways and talking to people. That's been a sort of a very personal challenge to deal with. On sort of the more sort of professional side, sort of the interaction we would have at conferences, discuss sort of science in different environments, brainstorm ideas, all of that is gone to some extent. A lot of my research has been supported over the years by the Department of Defense. And, we've always in some of those meetings in past years talked about what it would mean if some emerging viral or bacterial threat came about and there was not right tools to address it. And so while we never really did any active research to address that specific question, that has always been at the back of my mind. So, the broader question for our community as mechanical engineers, aerospace engineers, nuclear engineers, is to think about where we can use our specialized technical skills to address challenges for public health. It could be in sensing, development of new kinds of PPE. It could be on looking at the basic science for different tools. Healthcare tools are a very big deal. Ventilators were a big step for us to think about. And a ventilator is a mechanical device, right? So there was a lot of domains for us to think about from just this one disease. I think there are opportunities across the healthcare domain to think about this.
So then a question came in, a little unique, but we'll ask it. What would be your recommendation for students who might be looking for a job after graduation right now, kind of given the current situation? Do you have any thoughts or opinions on that matter?
I guess, the first thought that I would have is, as you're graduating, is great job on your graduation and all the hard work you've put in. And, you have the Ohio State brand behind you. So, you're going to do well in your life and career, and my best wishes for that. And, these are hard times. Businesses are hurting. So, industry jobs are hard right now. There are companies that are hiring, especially in the healthcare domain. Reach out to some of those avenues. Other large corporations like Procter & Gamble are hiring. You may want to sort of think and tailor your search to who's hiring. Another domain might be to think about, various government agencies that require smart and qualified people at lots of different levels. And a third choice, if you're intellectually curious and are excited about the science and the technology and trying to figure out how we can make a positive impact on our world, is to think about graduate school. I think that's a perfectly viable career path. I'm not saying that just as a professor, and we always like to have talent in our groups, but I think, when I made my choice after graduation to go to graduate school, it was not an easy decision. And I can also say that, I would not do it any other way. So it was a very rewarding experience. And I think that got me here. And, we've been able to do some pretty phenomenal science and technology over the years. So those would be my suggestions. I don't know if that answers the question, but that'd be what I'd say.
Final question that came up, and this is just kind of a side question. You had mentioned that you had done some water research in the past as far as clean water and things like that. Do you want to talk a little bit about that since you have a minute or two still?
Sure. Our focus on water research was in really sort of two domains. First was looking at sensing of water quality to figure out what the water can be used for. And then the second part was looking at, desalinating or cleaning the water in terms of treatment. And, we developed several new kinds of membranes to filter out water extremely efficiently. We've put those in systems. There is a I am allowed to disclose this, that, a few years ago, we had a fairly large DARPA program on water treatment where, and this was born out of the need for the supply lines actually being interrupted back in Iraq and Afghanistan by people just throwing rocks at armies, what was at the time called the lightweight purifier, which was a massive structure that sat on the back of a flatbed truck. And the enemy would just come in, throw rocks at it, poke holes in it, and now you have a platoon of 600 soldiers that didn't have water with them. And the soldiers in the field at the time were literally trading guns and their bullets to get bottled water and other water resources from local residents. So, you can imagine it was a logistical nightmare for the Army. So DARPA came out with this program on developing a small water unit that would fit in the back of a Humvee and would be powered by a bunch of truck batteries. So it would be in an armored vehicle and keep the soldiers safe and their water supply safe. And I think, Ohio State played a pretty cool role in that project. And we developed that system and handed over to the Army, just a couple of years back. And as far as I know, it's doing quite well. So, those are some of the projects. And those, again, go back to that first idea that I promised, was that, we look at the science at the micro and nanoscale for transport processes, and then try to build a device and a system, understanding that science, and then taking it all the way, if possible, to a translational stage.